name is Sam Kruzek. I'm an undergraduate student at the University of Colorado Boulder, and the project I'm working on is looking at scaly leg mite infections in blackcap and mountain chickadees on the Front Range. The Taylor Lab, which I work within, has a system of nest boxes from Boulder to Niwot Ridge that they use to study the hybridization of these two species. They've been monitoring and banding birds from those boxes since 2019, and since then they've noticed that around 10 to 15% of the birds had scabby, swollen lesions on their legs. They also noticed the majority of these infections are at about 2,500 meter elevation. I heard about this and decided to investigate it for my honors thesis project. As I started researching it, I discovered that these lesions are caused by an ectoparasite called scaly leg mites, which live in and feed on the skin and the legs of these birds. They are obligate parasites with the entirety of their life cycle existing on these birds. They were recorded in mountain chickadees in Canada once in 1966, but this is the first time these mites have been recorded in mountain chickadees in the US. This is also the first time they have ever been recorded in blackcap chickadees. During the field season, I collected samples from the birds we caught that had signs of mite infection, which I've done a couple of things with. First, working with insect biologists at CU Boulder, I looked at some mites under a microscope to get some measurements of their external morphology. Using that, I'm able to say the species we were seeing is likely Nematocopptes jamaicensis. This makes sense, as this was the species that had infected mountain chickadees in Canada. I've also extracted the DNA from these mites and will sequence the COI gene to compare to past sequences to see what Nematocopptes species these mites align with genetically. After identifying which species is infecting these birds, I of course would like to know how the infection affects the chickadees themselves. One thing we do know is that this infection can lead to the loss of legs. The infection causes swelling and crusting of the legs, so if a bird has a metal band on, it can restrict blood flow and lead to the loss of their legs. We have never banded any birds with any abnormal legs, but they can become infected after we band them, unfortunately leading to this. I would also like to look at the effect of infection on fitness. I will be investigating the effect of infection on clutch size, nestling number, and nestling body condition, as well as the body condition of the birds themselves, using data from 2019 until this year. There have been a few cases in Boulder and at higher elevation near the University of Colorado Mountain Research Station, but the majority of these infections are at our mid-elevation sites at about 2,500 meters. I want to narrow down what might be contributing to that cluster, so I'm going to analyze the effect of climate on infection rate, as well as adjacent habitat types. Past studies have found that drier habitats may increase infection rates, which may explain this, so I will look at the effects of temperature, precipitation, and habitat type on the prevalence of infection. Our data is limited to 2019 forward, and I was curious as to when these infections started, so I visited the University of Colorado Boulder Museum and the Denver Natural History Museum to see if any of the chickadees in their collection have signs of mites. I didn't see any signs, but I don't necessarily take this to mean that there were no birds with mites. This could be because the legs might change after they're processed for the collections. Also, further into the past, museums have rejected individuals with any sort of pathology, which could be contributing to this sampling bias. Understanding how scaly leg mite infections are affecting these birds, how the infection rates are changing over time, and how climate is affecting infection rates can inform whether conservation efforts may be necessary and will contribute to our understanding of this ectoparasite.